Thanks, Chris, for that really comprehensive picture from Pfizer. So I'd like to ask all the speakers to come and join me up here for a panel discussion. You guys have been perfect time-wise. We have half an hour to have this dialogue. And I think what you're seeing here is a really interesting mix of different stakeholders: nonprofit, academia, clinical, preclinical, and industry. It's a pretty good group of people to have a discussion with. And before we begin, I just want to sort of set the stage a little bit. You know, the data that you've heard presented around clinical trial programs, you have to ask a fundamental question. Are there too many clinical trials? And depending on whether you answer that yes or no, flows some other potential discussions. And so that's a question I'd like to, present to this group. And then the second question is enough effort being made preclinically to try and solve problems before we actually, or come up with plans for clinical evaluation that doesn't require the sort of spaghetti on the wall approach? And then the third question is, are we collaborating enough across the different stakeholders? So those are just general questions that I'd appreciate some responses to, but perhaps we can begin with a specific question.
So my question is, I like the...
Perhaps if you wouldn't mind popping to the microphone and just telling us where you come from, and hopefully we can hear you.
Great, thanks. So I don't know if everybody can hear me. But really great presentations. I love the distinction that Dr. Clive made between logical and scientific, modes of, of thinking in between in trying to come up with these combinations. But one of the problems we have as clinical investigators is that, there's a lot of talk about doing randomized phase 2 trials. But I think the problem is that you can't really, as the statisticians tell you, you can't really tell the differences between different arms in a randomized phase 2 trial. It's an exploratory way of looking at things, and you really dilute the power to tell the difference. So I think that brings back the issue of how exactly we power phase 2 trials, which I think are the major vehicle for us to discover mechanisms of actions and validate targets. And I think is just open it to everybody to tell us what they think about, should we be looking at novel trial designs? Should we just do a bunch more trials? Thanks.
Thanks for the question. So I hope you could be here after lunch, because there's a whole section on clinical trials and potential, new approaches. But just quickly to have a response there, perhaps, Ramy, do you have a comment on that?
Yeah, so I think randomized phase 2 studies is one option, but there's a lot more that can be done. And obviously, again, there was a session in the afternoon, but adaptive study design is an area that FDA and others have been promoting and asking companies to really consider the option of an adaptive design. Doing randomized, but not to randomize against standard of care, but randomizing different combinations so that you can pick one that's effective compared to other agents. And I think spending more time in the phase 1 study before jumping into the randomized study, relying on a clinical endpoint obviously is great, but there's a lot that can be learned from the preclinical work, the phase 1 data when it comes to generating biomarker data and really generating mechanistic data that informs whether your agents are synergistic or just additive or even no benefit when you combine them.
Yeah, please.
So I like to make a pitch for another model in terms of accelerated clinical development, which is the neoadjuvant model. Was mentioned by somebody this morning, I forget who, but I think I'm a real big fan of that. We've been successful in bladder cancer and in doing that, and really the paradigm of really understanding it, especially given the need to really understand mechanism and also for high throughput. Bladder cancer, if you look at treatment and then the patient undergoes a radical cystectomy, you have tissues before and after. You can look at mechanisms, you can look at biomarkers, and all that happens in about 6 to 8 weeks, depending on the protocols. It's relatively rapid, and the surrogate endpoint is no tumor in the specimen in the cystectomy, but of course you have tumors in the ones that don't respond, which is highly informative. So that model, and its bladder's not the only one, I just know that one the best. I think that I'm making a pitch for that kind of model to really study and do high throughput and cycle through a lot of combinations, timing, very sequencing. It's a rich platform to do that.
Perhaps, yeah, Rory, please. Yeah.
Two comments. One, the adaptive trials, I think those do have a lot of promise, and they're good for patients too, because then you're learning in real time, and patients are more likely to get combinations that benefit them. The problem is you have to have enough data to develop your prior probability and to know which biomarkers to use. But that's going to require, collaboration. And if there are too many trials, let's get all those data from all those trials into one database so that we can do that. The second thing is, I like the neoadjuvant approach. In certain tumor types, that's hard to do. Even the adjuvant approach could perhaps be used. We don't have to see actual resist responses anymore. I think we'll talk about that this afternoon. But ctDNA, just some evidence that there's some activity, just so we can have a little bit more information to tweak with the different models we heard about to say which combos should go forward.
I just perhaps want to make a case for randomized phase 2 studies or even randomized phase 1b studies. We actually got palbociclib approved on a randomized phase 2 study, and we currently have glasdegib, smoothant inhibitor, under priority review with the FDA for AML on a very tiny, small, 110 patients randomized phase 2 study. So it all depends how the study is powered in terms of how meaningful is the difference. We are looking with these combinations for meaningful differences. The reason is these are going to be 2 or 3 different medicines as being combined. It has to be meaningfully better than a single agent because of the additional toxicity burden and, of course, pricing and access burden. So I think detecting signals in a randomized experience in a small study, for example, at Genentech, the Morpheus study, which is a kind of adaptive study, but it's randomized to really determine early on those signals, I think is important.
Thanks. Next question, and please just introduce yourself.
Fabrice Milauskas, University of Chicago. 2 questions. Number one, the costs of care are of big concern to patients and to the healthcare system as a whole. And obviously the cost of combination therapy may be more extreme than the cost of monotherapy. Is there a role for considering the cost of care and prioritizing combinations? For example, combining with off-patent agents like many chemotherapies, or with already approved agents which are likely to be going off patent very soon, and in some countries with weaker intellectual property protection around the world may already be off patent or have generic equivalents. So one question. The other question is, Emmett was suggesting that the rise in number of trials is really driven by the investment community. If that's the case, investment moves in cycles, and there may soon be a correction in the stock market, which would greatly reduce the amount of venture capital, the amount of IPOs, and investment flowing into trials. If we saw a big drop in the number of trials, are we ready for that as an immunological community, as the cancer community? Cancer science will continue to progress, but potentially the funding for it might, within a year or 2, drop substantially.
Well, it depends on whether you're a spaghetti thrower or not, I guess, whether you think that's a good thing or a bad thing. The spaghetti throwing has had some virtues and has produced some results that were not those that a large number of people predicted. The chemotherapy thing was, the lung team feels, pretty beaten up over the last several years about the comments from people from immunology. And yet, that's an unequivocally positive trial. And by the way, it speaks to your other point. Those are off-patent agents. Those are economically viable. I think some of this, and I'll speak as a Massachusetts liberal who spent a lot of time at Harvard University, and I'll just note that there's some virtues in capitalism.
Just a comment on the question of, is it the investment community? I must say, Emmett, I have to take issue. This is driven by patient need. There's such a high unmet need in cancer that every effort to try and improve an outcome is a laudable effort. And so I would argue that actually this is really what it's all about. And
of course, Roger, but the ability to sustain that addressing.
The capability, that's true.
And many of these studies are driven by academic sites as well, by academic hospitals. So I don't think there's too many studies. I think there's A redundancy in some studies, that's an issue, which could be addressed by big data and by databases, that the same study is not repeated. But if we look globally, less than 1% of patients enter cancer clinical trials. In many regions of the United States, it will be less than 5%. So I think having patients having access to clinical trials is a bigger issue than too many clinical trials.
Yes, Roy, would you like to respond to that as well?
I would agree. And if you look worldwide, there are so many patients worldwide not getting these agents right now. U.S. Even with all the problems, it's better than most. I guess the big question would be: are all PD-1 and PD-L1 agents the same? So there's so many of them now, and now with biosimilars coming down the pike, that's going to be the big issue, differentiating between the agents. And I've been struck. A phase 1 comes down the pike, and it's a new agent from company X, and they have a new PD-L1 inhibitor that hasn't even gone through testing yet, and they're combining that together. And so I think that's a question for the panel. It's hard to say. I guess the FDA might have some of those data. Maybe we'll hear a little bit about that later, comparing them. But I think that's an issue.
So I think the point that you mentioned is going to be a major core of progress, to be honest. The economic part of it is not a non-simple thing. And I agree with Emmett to a large degree. At the end of the day, you're not going to be able to do clinical trials if you don't have people that are putting money for those clinical trials. That takes us to two things. One is the more clinical trial potentially to fail, the less money will get there. And the second thing is that the cycle is going to be determining how to move forward. So that takes us to the second point, which is the expense. The expense is going to be high. So the question is, yes, can we actually start thinking of repurposing drugs? Can we start thinking of natural products? And as a matter of fact, natural products are probably very much well suited within the immunology field to take over lots of those kind of approaches, not to minimize the work of pharmaceuticals and the need for the development, but that might provide tools that are way less expensive, because it's going to be very difficult to maintain a field that a patient is going to cost $400,000 or $200,000 per year for a long time to come at the end of the day, particularly if we maintain 15, 20% of response, right? So these are the things that we really need to think about.
George Weiner from Iowa. So a challenge in small studies, both supported by industry and academia, is storing quality biospecimens for future biomarkers. And as we do all these small studies, the opportunity to learn from them is being lost because we're not doing that in a consistent and reliable way. So huge numbers of patients going on these trials. How do we be sure we have the biospecimens to learn from them when we identify future biomarkers we want to assess?
That's a great question. And it comes to the issue of, is there something we should already know that we don't because we're sitting on a mountain of data we've not yet interrogated? So, Ramy, do you have some comments on that?
Yeah, I mean, one area is studies, especially the ones that had negative outcomes, because those will not be the top priority for the private sector to go back and analyze the data. So I think this is a very rich data set that other groups, whether it's cooperative groups, whether it's nonprofit, whether it's government, if we can have access to samples that were collected from those studies, we have clinical outcomes. There is money outside of the investment work that will be available so that we can learn from negative studies, which again, I think it's an area that is going to get bigger because there will be more and more studies now that are not having an early signal from the phase 1 studies, and they will be shelved. There are lots of initiatives now. PACT is a group that started looking at standardizing how samples are being collected and standardizing the way that analysis is being done in immunotherapy studies and other studies. And then there's work that's being done with the Parker Institute on a limited number of centers to introduce this standardization and really trying to learn from the biospecimens that are being collected on clinical trials.
I would just comment, when the budget's over by 10%, what goes first? Storing the biospecimens for future use. And I think somehow or another we have to get beyond that.
I just add, it's not only storing, it's using them. So, like the chemotherapy example, that Emmett was bringing up, huge, but has anyone looked yet at the T effector in memory cells? What's going on in the blood? I'm sure some of the companies are, but you have to ask a hypothesis-based question. And, even at one institution, I think we all have that experience. It's hard to get the tumor banks working in every tumor type and storing in routine ways. But there's so many hypothesis-based questions we could ask, first in local areas, but then coming together in a collaborative way. And I guess that's where the institutes and other groups come together.
I might offer a couple of practical comments. So the freezer space isn't the issue. The issue is, where is the specimen? What was the consent? What are you going to do with that specimen that's not going to be, I'm going to do every single test known to mankind on it so that you immediately move outside multiplicity control? And I'll, for all that there are virtues in our direct collaborations company to company, that actually is the harder place to solve from the standpoint of biomarkers, because now when you get into exploratory stuff, both companies are going to get a little nervous about intellectual property of things that are discovered in that kind of exploratory set. So that's a comparison. I think that is a place probably where an extension over into something like the Parker Institute might make it easier on these collaborative studies, where the intellectual property, you both give it up.
Right. Yeah, we do have a session tomorrow morning talking about pre-competitive collaboration and data sharing, which is obviously a key area. It seems such a pity that there could be specimens sitting somewhere, or even information sitting somewhere, that others are not aware of.
And I also just wanted to point out, industry is very interested in the negative studies. We don't want to repeat negative studies. We're probably more interested in the negative studies than in the positive studies, because you can get cleaner answers, and it's telling you what you did wrong. So I wouldn't assume that that's going to be an academic purview only.
Dan?
Thank you, Dan Chen, Genentech. So with all of the discussion this morning about throwing spaghetti against the walls, I would hope that for the future of combination immunotherapy, that's really not the direction that we go in. What I think we've seen is a proliferation of a lot of studies, duplication of the same scientific hypothesis repeatedly, and I actually question how much we've actually learned, at least in the combination immunotherapy space, from throwing spaghetti against the wall. And in fact, if you look at the positive results that are now coming out in phase 3, I believe all the positive combination immunotherapy trials have been the combination of two active agents. And so when you're combining two active agents in a disease, I wouldn't really refer to that as spaghetti against the wall. You know these drugs work. Now you want to understand if they combine well together. And so I do wonder whether, as we move into the future of combination therapy, that we can really start to more rapidly prioritize and push forward those studies where we have multiple active agents that we're combining together. And that doesn't mean that you can't have a combination where one drug doesn't have single agent activity, but perhaps that should be the minority of the studies we have moving forward.
That's a great point. Chris, do you have a comment on that?
Yeah, that's a very, very good point. So I think the first lesson for all of us after epicadostat specifically is that we probably have to combine two active agents. I think the next big challenge is something that Emmett showed, this recent Cell paper from Sorger and his colleagues at MIT, I believe, with Harvard, where we can have two active agents, but when you combine it, there may be independence, meaning one population benefit from one and a small population benefit from the other. And there's very few patients actually benefiting from both agents. And actually in their Cell paper, they use Ipi/Nivo as an example, when many patients, in the case of many patients, actually there's no combination effect, there's independence. So we don't want to expose patients to two drugs if they could benefit from one. And I think that's one of the next challenges to build into these studies, but a very good point.
I'm going to take a point on this one. I worry about this. And the reason I worry about this is because I believe that, again, if you do have biologic data, this is what should guide us. The mere fact that one drug does not work on its own in immunotherapy, in my opinion, that might not be enough not to combine it. I mean, case in point, we did vaccines for 20 years, and they never worked. We know that in combination, that might be completely different, especially with the neoantigens, et cetera. So I would be very careful putting a sentiment, because lots of people start thinking of this. I mean, lots of people start now saying, well, let's kill this drug. Some drugs might be very, very powerful in combination because the other drug is waiting for this change or this manipulation to happen to work better. And this is why I would say, again, we're not going to have enough patients to do all clinical trials. Clear. I mean, that's clear. So the question is, what can we depend on to start selecting? And if we have good biologic reason to think that those combinations, despite the fact that a drug did not work on its own, I think that might be a good reason to go forward. And I could give example after example where companies now, and you know that very well, Dan, from Genentech, starting to actually take on some of that strategy. Well, let's kill this drug because we don't have, and my worry is we're going to be losing very good drugs.
Hi, I'm Tanya Canigan from Genomic Expression, and I'm wondering, to what extent would you consider therapies that are given serially to be combinations? And I'm especially thinking about when immunotherapy is given after chemotherapy or radiation therapy, where that's known to, of course, potentially transform the tumor microenvironment. Should we really be thinking about those sorts of combinations as happening, even if they're not at the same time, or maybe even if the decisions are made independently?
Dan, do you have a comment? Dan, any thoughts?
Well, I have several thoughts, but I think the idea that you can reprogram the tumor microenvironment after chemotherapy and is very important because that microenvironment is not going to be the same when you do serial versus simultaneous, right, combinations. So I think the idea that you can extrapolate from a serial result to one with a combination, I wouldn't assume that's the case. Having said that, at least in our experience with the animal models that we've used, the serial combination and the sequential, the serial combination and the simultaneous combination seems to have similar outcomes. But those are models.
There's at least one example, which is Steve Hode's IPI then Nivo, Nivo then IPI, where they couldn't enhance activity. They could not enhance activity, but they never lost toxicity. So at least from the one experiment that's out there at a reasonable powered size, sequencing was not a good answer. Then historically, that's actually usually the first question we get in these sorts of panels. It's in everybody's mind. And my historical note is it took a long time before pediatric leukemia went over to sequencing as a solution. So I think we're a number of years away until we understand the basic principles of what you combine before we're going to really effectively think about sequencing. And that's just a historical observation.
I think one area where that needs to be looked at is with the chemotherapy, though. Perhaps, there are patients who can get the immunotherapy alone first and then follow with chemotherapy. And there are some single-arm trials that show that the chemotherapy response rates seem higher than that historical data. The problem there is if you don't know who's going to benefit or not at the beginning, if at least there are independent effects, you might need the chemotherapy early on to keep the patient stable enough to have the full benefits. But I think those are going to be studies in use that are going to have to be done. And some of the cooperative groups are beginning to do trials like that based on some of the chemo combo data.
And of course, the Pacific study is the best example for sequencing.
Right.
The AstraZeneca study with durvalumab post-chemoradiation.
Thanks, Roger. Martin Murphy from the CEO Roundtable on Cancer. What a terrific beginning to this National Academy meeting, as they all are good. This is a splendid beginning with the seven of you, the Magnificent Seven. Any of you see that in German, that movie? It's called Die glorreichenden Sieben. Sounds even more.
You're landing in the middle of the spaghetti, since that makes us a spaghetti Western, you're landing in the middle of the metaphor.
That's where I was going. Emma, that's exactly where I was going. But to address, Roger, your entreaty, that is, are we doing enough collaboration? I think, again, this session this morning is showing the tremendous collaborations that industry and academia and government have really instituted in the IO space, without question. But there's an area that requires, I would suggest, in addition to tumor sharing, an opportunity sharing. And you know, Chris, where I'm going with this, and that's data sharing. And I mean data at the individual patient level. Tomorrow we may hear, because I'll be talking about it if you're here, Project Datasphere as an initiative, a nonprofit initiative. Many of you are actively collaborating with this open access, a truly open access data sharing model. in which Rick Pastor last year issued a letter of support specifically for immuno-oncology trials where there should be this individual patient level data sharing, and there's not yet enough for clear reason. Second thing, just as an initiation for further comment, perhaps by you, Chris, in the sunitinib, where you now are really seeking to aggregate curated sunitinib control arm, again, the FDA with its prescience is encouraging, again, Project Datasphere to do small cell lung cancer comparator arm aggregation after curation to eliminate the need for that in small cell lung cancer. The same thing could, and I would submit should be done here. We can eliminate comparator arms by really good data sharing. Thanks, Roger, and I hope you take that one off. Thank you.
Perhaps the only thing to add is that we'll hear tomorrow more from Amy from Flatiron, I believe, to expand on the potential use of synthetic arms and where we are with synthetic arms and whether it's actually looking at historical real-world data or in real time doing a randomized study where patients are being randomized in the real world for each study in your... It's a very good point. Thanks.
Yeah, you know, I mean, a lot of the early sort of signals are sample size issues, really, aren't they? I mean, you could do a very large trial, and it could be a single-arm trial, but it will tell you, it will give you the confidence intervals around the result. And if you have a similar, close to, or almost contemporaneous, one could imagine making that sort of comparison without, you know, suffering the tyranny of small numbers.
Roger, you asked global metaphoric questions, which is, are we doing enough, too few, too many? Is this Goldilocks number of trials? I mean, you know, given the job that you assigned me, my answer is sort of intrinsic, and actually you also gave another reason for the answer, which is there's plenty of unmet medical need. Why wouldn't we be doing these trials? So I don't know if any of us would think that we should stop this pace. There is unmet medical need. We are answering questions. The majority of the trials are positive in these phase ones. So why wouldn't we be doing this?
I think the question is, well, I mean, others may disagree, but one of the concerns is we're doing a lot, but are we in fact getting the maximum value out of the effort? So that's Roy, you have some thoughts?
Yes, there are too many, but also too few, because, you know, we need trials based on the science. And I think if all these data from all the trials came together, and we understood resistance mechanisms, I think that's going to be key, because now what we're seeing is everyone's getting these agents in the approved setting. And then they're coming in and they're refractory, and they're going to phase 1 clinics, and they're getting different trials. And the data for what to use in the refractory setting are really small. The preclinical models, as we heard, are okay, but really not adequate. And the studies are still very few. So I think a controlled effort, you know, we need many trials in this setting, but it needs to be based on biomarkers. At least if we're not putting people on trials based on biomarker, we should at least be obtaining the data so that we can learn in the future. But, you know, what strikes me is that many of these trials that are designed with hypotheses in mind, the trial's done without using the biomarker. So I think we just need to have to be as smart as we can. But no, I don't think that there are that many trials that shouldn't be done, especially since we need to get these out to the community and to patients throughout the United States, throughout the world.
Hi, Jeff Allen, Friends of Cancer Research. Thank you for all of the presentations. I was a little surprised that only a couple of you mentioned within the case studies the presence of additional toxicities. So I guess just a question. I know some of the case studies were exploratory, but and looking for signs of efficacy, but it seems with synergistic effect could come synergistic toxicity. So I'm just curious as to what your thoughts are moving forward. How big of a problem will toxicity be, both in looking at eventual efficacy as well as implementation of successful candidates into more community-based settings?
I want to jump quickly to that because that's another virtue to the orthologous thinking of independent action. So again, how were the original pediatric trials designed? They were designed to have orthologous mechanisms precisely to avoid shared toxicities. So moving away from two like agents avoids that toxicity, and also it avoids coincident resistance mechanisms. So again, in the history of combination therapy, those are our traditional approaches to answering that question.
Perhaps just to add, that's a good point, Jeff. Specifically for some of the TKIs, we now know of a number of the tyrosine kinase inhibitors where if you combine it with a checkpoint, it's almost as the immune system protect against some of the side effects of a TKI. So, for example, some of these TKIs in lung cancer give pneumonitis. If you combine it with immune checkpoint, it looks like the lung cancer, the lung toxicity is increased. So I think that's important, and it will be important to detect that early on before embarking on a bigger study, obviously. As we saw recently with the studies with lenalidomide, with multiple myeloma, where a number of the late-stage studies had to be stopped or changed because of the detection of toxicity.
Well, just a quick note also, of the trials that we're running, I'm in the neighborhood of 90-ish. 90% of the agents are going in at full dose of both agents. It's actually the combination of the PD-1s, I think, is amongst their really astounding virtues as individual agents. And they combine well with limited toxicities. And interestingly, there is a toxicity which is shared amongst them, which is not necessarily noted. But LFT abnormalities when the combination agent seems to be the thing that sticks out as the unusual toxicity.
Can I just make one comment here, and back to the question about the number of clinical trials? So the fact that we have agents that are well-behaved and combine well with others should not be the reason why we're combining them, because just the fact that we did not see toxicities in a study, this is excluding the patient from the next study, because many of the potential effective combinations will exclude a patient who receives prior PD-1. So I think the fact that it's tolerated, the fact that there's some early signal of activity, we still need to be very careful with which combinations are we testing.
And some of these side effects do occur later on, and again, using the example of multiple myeloma with lenalidomide, that was not detected in the early phase 1B and with the first 20, 30 patients. It was only later on during the study. So just some caution.
Right. But I mean, the mantra in drug development is we should commit to developing safe and effective combinations. I think that's risk-benefit.
Meaningfully effective.
Yes. Naya, please.
Hi, Naya Rizvi, Columbia University. So, when we were flooded with all these new IO agents maybe 10 years ago, I think that a lot of us felt that there needed to be single agent activity for a compound to move forward. And then we saw the combination data and we went, okay, this is cool. So maybe we don't need single agent data. Maybe we should be circling back to requiring some single agent data in a tumor type for a compound to move forward.
So that's a safe place to be. I think that's the point that Dan Chen was making, is if you choose, based on what Emmett's describing as the sort of orthogonal action, if you have two active agents just at first principles, independent mechanism, you can make an argument for the combination. And so the chances of success are much higher when both agents are active. But to Samir's point, that would then preclude some, potentially very important biology that we would be ignoring. Other comments?
What about a compromise plan where there was a pharmacodynamic marker or something that at least showed you were having a biologic effect? That might be good, right?
Absolutely.
Sort of inevitably.
But that's the point. Actually, this is the issue that we really need to learn more about, Nair, is are we using the drugs in the capacity that we need to use a drug in? OX40, for example, is the ultimate example. I think OX40 as by itself might not actually provide you with some data that would be of positive outcome, but did we use it in the wrong way by using it with PD-1, or it's better to use it with something else? And we learn the biology of this. And when we learn it and we understand it, then we use it for that. Because I actually bet publicly here that OX40 insert combination is going to be whopping good. So I think we need to be really very careful on killing some of those drugs or using them in the wrong way. This is why we really need more science and understand the biology and understand if that translates into human or not.
Being an OX40, I hope he's right.
Okay. Well, I think we're going to draw this session to an end. It's just for me to thank all of the presenters and participating in the panel, particularly to thank Samir, my co-chair, for a really good beginning for this meeting. So thank you all. We have a break, and I think we reconvene at 10:45. Thank you.